Clear? So, good afternoon, everybody. Today we discuss the development of endosperm. So, the process of double fertilization, which is unique to the flowering plant. Double fertilization is a specific character you can see in the flowering plants only. The first fertilization or syngamy, which results in the embryo development, which is a feature shared by the non-flowering plant. It is a specific feature. The second fertilization, usually designated, we can see as a triple fission, which is the exclusive specific feature in the flowering plants, which is product to the endosperm is correct only in the angiosperm. Okay, double fertilization is the specific type of the triple fission in the angiosperm. So, historically, the second male gamete involved in the triple fission is initially in the lilium and the fritillaria by the Navashi. So after the double fertilization was reported in the larger number of angiosperms. Okay. The fission product of one sperm with the secondary nuclei results in the formation of the primary endosperm nuclei. The nuclei is divided through the mitosis of this nucleus and daughter nuclei constitute the endosperm. So it is the families of Orchidaceae, Podostomiaceae, and Traphaceae, which is the characterized by the absence of endosperm. What are there? Orchidaceae, Podostomiaceae, and Traphaceae. We cannot see the endosperm here. The endosperm being the pool of reserve food material. This is the reserve food material for the development of the embryo, growth and germination of the embryo. The endosperm may be either be consumed by the developing embryo, example peas and beans and pulses. If it is not utilized by the embryo, which is result as a persistent immature seeds, that is the cereals, castor, bean, and coconut. Okay, some embryos are to develop the nutrition from the endosperm. We cannot observe the result as a persistent embryo. So there is cereals, castor, and beans and coconut. Next one is the development of endosperm. So here the development of endosperm, here you can see the so here the development of endosperm means based on the mode of the development, the endosperm is classified into three types are there. Means the development based on the development. Here endosperms are classified into three types. That is the nuclear endosperm, cellular endosperm, and helobial endosperm. Okay, these three types. First one is the nuclear and cellular and helobial type. Here the nuclear type. In this nuclear type, the first division of the primary endosperm nucleus and several of the following divisions are not followed by the wall formation. Okay, so here wall formation is not observed. So as a result, the free nuclei either aggregated in the micropylar or callagel region became distributed in the film of the cytoplasm around the central vacuole. So here, what happened here? After the after the division, means several divisions. After the several division, we cannot see the wall formation. The endosperms remain free nuclear throughout the till the endosperm is completely consumed by the developing embryo. In the, for example, what is that one? Oxyspora and Limnanthes. So these are the, so wall formation occurs in the different stages of the development in different species. So here, for example, four nucleate endosperm in Caffea arabica. So here, 16 to 18 nucleate stage in Tithonia, and after the formation of around 1000 free nucleate in the anemone. So here, depending on the different species, here having the free nucleus in the endosperm. The wall formation is the centripetal, or it is the initiated around the proembryo at micropylar and proceed the chalagal region. In the most plants, wall formation once initiated to continue until there is are no free nuclei. The cells divide until the maturity of the seed and get filled with the reserve food. In a few families like Cruciferaceae, Cucurbitaceae, Leguminaceae, and Proteaceae, here wall formation does not proceed behind the central region of the after initiation. So what happened here? In many plants, the species structure are going on the endosperm, either one or both poles of the endosperm sacs are there, embryo sacs are there. So these formations present in the morphological and histological distinctiveness from the remainder of the cell tissues are believed to aid in the nutrition of the developing embryo and seeds. So in families, for example, Proteaceae, the Proteaceae, most of the endosperm nuclei are distributed in the upper portion of the embryo sac. So the cell formation is restricted to the upper portion, while the lower portion of the sac remains free nuclear. So here, what happened? In another member of the Proteaceae, Grevillea, Grevillea robusta, the lower sinusotic portion of the endosperm grows in the form of a colloid and tubular worm-like structure, have been designated as a vermiform appendage in Grevillea species. So here, what happened here? You can see the a vermiform appendage like structure is there. It is served as a hastoриum of a very aggressive type. So later, the appendage becomes a portionated into smaller units so that the cell formed in this way constitute a kind of secondary endosperm tissue. Okay, so here, just like to form as a, okay, this is the, see here, and the so dissection and the microtome sections, they remove many interesting types of collagen endosperm. Okay, So when the pre-embryo is the globular stage, the upper broad part is the endosperm and became a cellular, and it is the designated as the endosperm proper. The lower portion here, the pre-nuclear portion is elongated still uh, in some plants is the frequently reached down the thalassal region here. To begin with, the endosperm hastorum is cynocytic and is many cucurbits is remains so throughout. In Citrullus, it became a segmented into the multinucleate chambers, which later became a subdivided into multinucleate cells. So in Coccinia, the hastorum usually became the cellular, but sometimes it is the apical part remains the cynocytic condition is there. So in Mammodica, this is the Mammodica, the wall formation takes place in occasionally. Wall formation is also there in the Mammodica carentia. So the tip of the here, so the tip of the hastoria in Cucurbita and Cucumis, they became a swallow. It is a swallow. The longest endosperm hastorum is reported in the Echinocystis lobata, where it is measure up to 16 millimeters in length. Occasionally, the peripheral cells of the lower region of the endosperm proper plug out during the later stages on the development of the seed. So in loofah and Melothria, so such bulbs are usually referred as the secondary hastoria. So here what happened in thalassal endosperm, the hastorum is also recorded in Leguminaceae. Okay, in Stylosanthes, the cell formation is absent. The cell formation is restricted in the region immediately around the embryo, while in other it is restricted to the upper half to third of the embryo sac. The lower half of the endosperm is remains free nuclear, assumes the vesicular contour that contain hypoperipheral nucleus and formation as hastoria. The shape and size of the hastorim it is the varies in many tags. In Dicrostachys and Mimosa, Crotloria, and other some species of the Acacia assume the tubular form with the vesicular tip. So in Acacia and Mimosidae and Acacia occidentalis, the anther bulbar pre-nuclear portion just below the cellular zone of the endosperm. So this is the so these astoria usually remain the cynocytic condition, but in the Cymopsis and Cymopsis too, So the Asherams usually remain the synesthetic condition, but in the Cymopsis and Desmodium, the valve formation is eventually takes place in the tubular processes. In scleria, sorry, in scleria, the presence of both micropolar and chalazal endosperm cluster has been reported. You can see in the both chalazal and micropolar astoria we can see in the scleria. At the two nucleate stage, the tubular extension is formed at the micropolar end and embryo site. It remains in the nucleate with the catty cytoplasm and acts as a hastoria. The centripetal wall formation is restricted only to the upper region of the endosperm. So here the chalazal portion remains synocytic and they function as a hastoria. Okay. So the correlation between the size of the fruit and activity of endosperm is exemplified as a coccus nucifera. The primary endosperm nucleus is undergoes a number of free nuclear division by the time of the fruit became here 50 millimeter long. The embryo sac is filled with the clear fluid. It contains many free nuclei of the various sizes of the cytoplasm and usually referred as a liquid syncytium. When the fruit is about 100 millimeter long, the suspension shows the long free nuclei. The several cells with a variable number of nuclei. So these cells and the free nuclei gradually settle at the periphery of the cavity and layer of the cellular endosome make their appearance, which forms the coconut meat. We know that one. Due to the further divisions of these cells, quantity of the cellular endosperm increases. In mature coconut, the liquid endosperm became milky and divide of many prenuclear nucleus are present. The development of the endosperm is the areca, comfortable to them in the coconut. Okay. So next one is the endosperm nucleus. So another noteworthy is the feature of the nuclear type of the endosperm is formation of the enucleate and nucleate cytoplasmic nodules are present. So here enucleate nodules have been reported in the passiflora. So in passiflora, there is the nucleus and some cucurbitaceae member. The nucleate nodules have been reported in the capsula, calyx is also, where the both enucleate and nucleate nodules occur in the Carica papaya by the thing in 1976. Okay, So this nodal distribution of endosperm nuclei is the periphery and prevents their accumulation around the zygote, so which otherwise the causes in endocrine to the growth of the embryo. The nodular cytoplasm is rich in various organelles. The nodules may either merge with the endosperm proper or degenerate. So next one is the cellular endosperm. Okay. So in this type of the division of the primary endosperm, nucleus and subsequent nuclear divisions are followed by regular cells by the cell wall formation. The latter may be followed by the transverse valve, resulting the linear rows of the cells in the in Custody phylum. Okay, linear lines are formed between the cellular and in hymenthermum, the endosperm nucleus undergoes the division followed by the transfer valve. The two cells undergo one or more transfer division, resulting the linear row of the four cells. Later on, the longitudinal division occur in the micropylal cells of the endosperm, making the biseriate condition. Gradually, these longitudinal divisions are extended to chalazial endosperm. The cells of the endosperm cells are also became a biseriate. See here, these biseriate condition. So further division in these cells occur in the all planes, resulting in the massive endosperm tissues are formed. So the formation of the hastoria is very common feature in this type of the endosperm, and it is more varied than the nuclear type. The hastoria may be micropylar or chalazial, otherwise both. This is the characteristic feature of the majority of the sympatalis family. The usually for the obtaining the nutrient and transporting them to the developing endosperm. The asteria grows in the direction of the tissue rich in the food reserve. So the micropylar asteria are reported in the impatiens and hydrocera. A two-celled chalazial hastoриum is developed in the Mongolia. The first division of the primary endosperm nucleus is followed by the transverse wall, resulting into the chambers of almost equal size. So the divisions in the micropylar chambers are rapid into all directions. The chalazial chamber is divided transversely and at the comparatively slow rate. So this results in the tail-like chalazial portion attached to the more massive tissues at the micropylar end. So here further divisions occur in the upper part of the tail. Micropylar, the upper tail and the endosperm. The basal two or three cells of this tail elongated to form the osteria, which is penetrate to the chalazial part of the nucellus. In hexocarpus, the division of the endosperm nucleus is by a transverse wall. Okay. Of these, rhetoric. Here, of these two cells, thus produce to the lower divide vertically, whereas the upper segment is transverse. So the two cells of the lower tied to the should not divide further. Okay. So these two cells of lower tried but is elongated considerably and produce many tubular processes that they serve as a efficient astoria. In iodina, the chalazal hastorum is the aggressive and profusely branched. The hastorum is actually formed before the fertilization. The chalazal end of the fertilized embryo sac forms an extensive in the cachium. The lower end of the extends to the placenta and the branches. After fertilization, the division of the primary endosperm nuclear is followed by the transverse proportion of the central cell. The endosperm proper is derived from the micropellar chamber alone. So here, the chalazal chamber function and an aggressive uninucleate hastoria. The nuclear migrates into the caecum and becomes the much hypertrophied. Preface the branching at the free end gives the hastoриum is correlated appearances are there. So in some families of acanthaceae, the acanthaceae member, the endosperm developed into the cellular type. The first division of the primary endosperm nuclear cut of the a small chalazal hastoриum. So the micropellar cells is divided, the transverse to give rise to the daughter cell, of which the upper becomes a binucleate and form the micropellar hastoria. The central cell, which is largest, form a sac-like outer growth where the several free nuclear division. A large vacuole appears in the center of the cells and nuclei become distributed in the periphery layer of the cytoplasm. Okay. The endosperm proper is formed chiefly in the central chamber. It may be nuclear, incubator The ball formation takes place at the 32 nucleus stage in Berleria and Rullia, at the 64 nucleus stage in the Dipterocanthus and Rullia, at 128 cells, 110 nucleus stage in the Rullia, Acaciana, and Adathoda vasca. So here, if the wall formation is not complete, the free nuclear basal apparatus is formed below the cellular endosperm. Such basal apertures is seen in the Dipterocanthus and Adathoda. So, which is the aggressive and formation many multinucleate and enucleated figure-like process. The deviation from the general pattern of the endosperm development in the acanthaceae is followed by the Blepharis and Thunbergia. The linear three-cell stage present in all of the other members like families does not occur in these plants. They are also locked the chalazal hastoria. After the division of the primary endosperm nucleus, a micropellar chamber and the chalazal chamber are formed. So these forms grows into the hastoриum, whereas the latter gives rise to the clugia. This shows both micropellar and chalazal hastoria. The chalazal hastoриum is the initially binucleate, but the two nuclei fuse to form the uninucleate cell. The hastoриum grows laterally and upwardly, consuming the subepidermal cells. The micropellar ostotrium comprise the two nucleus cells. Both the both the micropellar and chalazal ostoria have also been reported in the Pneumophilia, Hydrophiliaceae family member of the three cells formed in the micropellar and chalazal cells develop into ostoria while the cell is division are conformed into the central cell resulting in the endosperm proper. The next one is the composite ostoria. The composite ostorium means composite endosperm. Here, a unique type of the endosperm development in the present in the Loranthaceae family member. There is a beginning to true ovules. All these embryo sacs in a ovary is close to each other. They develop occasionally, so they develop concurrently, and their tips separate to various heights in the style and stigma. So after fertilization, the primary endosperm nucleus moves to the basal portion of the embryo sac. So whereas the after fertilization, the primary endosperm nucleus moves to the both basal portion of the endosperm sac where it is divided. The endosperm forms simultaneously in the several embryo sacs in an ovary and they fuse to the form and composite endosperm. This is a feature so far they restricted only on this Lorenthaceae family. The only the exception in the Tutanthus, Tutanthus vulgaris, where the endosperm develops from the a single embryo sac. This is the composite structure is not formed. So another type, heliobale endosperm. The heliobale type of the endosperm is characterized by the division of the endosperm, the primary endosperm nucleus, which moves to the chalazal end and in the embryo sac, so which is give rise to the unequal chambers. That is, the micropellar is usually larger than the chalazal side. So free nuclear divisions occur in the micropellar chamber before the undergoes to the cellularization. By the contrast, the nuclear and the nucleus of the chalazal chamber either they remain undivided and either divide to few times. So here, chalazal chambers usually remain the synesthetic condition, but a few cases like phylidrim and gensus would get the partial into the cell. In Sparganium, here Sparganiaceae family, the primary endosperm nucleus lies near the antipodals and it is division results in the large micropellar and small chalazal chamber form. The wall formation is the micropellar chambers does not always precede the chalazal chamber. The endosperm cells became the multinucleate. So here, while the remaining the halobial type of endosperm and suggest Swami and Parameshwar, here some certain norms are there. So what are the norms here and what are the criteria here? The primary endosperm nucleus occupies the portion in the chalazal region have the embryo sac in the neighborhood of the antipodal. The second character is it is divided in situ. And third one is the micropellar chamber is the larger than the chalazal chamber and which is separated either by the wall formation or embryo formation. The fourth character is the nucleus of the micropellar chamber moves away from the wall and undergoes to the prenuclear division. Okay. So here what happened, according to the Swami and Parameswaran, the formation of the true holobial endosperm is confined only the monocotyledon families. If it is occur in the dicotyledon families, it is only the modification of the cellular and nuclear type of the endosperm. So finally, however, this is the typical holobial type of endosperm have been reported to observe in the dicotyledon families such as the Santalaceae and Saxifragaceae. Okay, so next one is the ruminate endosperm. What are the ruminate here? This is the ruminate endosperm here. Ruminated means folding, the folding endosperm. So in this mature endosperm is associated with the uneven the surface and irregular outline. It may belong to any one of the three types of developmentally, and this rumination starts at the latter stage. The rumination are caused by the localized activity of the seed coat or by endosperm itself. In the first category, which is the irregular inner surface of the seed coat may be formed due to unequal radial elongation of any one of the or only the layer of the seed coat. The second is define the irregular growth and unfolding of the seed coat, that is the ananasi. So in Myristica species and Cocolaba, the endosperm becomes ruminated due to its own activity. It begins to increase in the volume and simultaneously with the increase the volume of the seed. It soon observed as a nucellus and becomes into the direct contact with the seed coat. So during the further growth of the endosperm, the irregular inner surface of the seed coat makes it is ruminate. And if in other hand, andrographis species, the endosperm exhibits the unequal periphery activity during the late stage of its development and causes the seed coat to attain an irregular outline. On morphological basis, and Perias farm in 1962 is identified the seven types of what are there? Annona, Passiflora, Meristica, and Spigelia, Perbexum, and Cocolaba, Elytraria. These seven types is observed. In the all these types except Elytraria, the irregularities occur on the growth of the integument, which will bring out the rumination of the endosperm. So in the Erythraria type during the development of the seed, localized region in the periphery layers of the cellular endosperm shows active growth, which is became a ruminant. The seeds which are reveal a network of the lobal section. Many plants are termed as the labyrinth seeds. So what is that one? Labyrinth seeds. This structure may be either due to the rumination of the endosperm or lobing or folding of the cotyledons. In seeds, thus, when the lobing and folding of the cotyledons occur together with the rumination. So here, the labyrinth structure is very prominent. So other species by the labyrinth seeds are not made within the convolution. Okay. So here The ruminant endosperm could be a ancestral character still occurring in the present day state belongs to both to advanced and primitive taxa. The cellularization of endosperm. Cellularization of the వస్తామని చెప్పపోయినావు right. In angiosperms, the nuclear and halobil types of endosperms have been pre-nuclear phase. The wall formation is synesthetic endosperm gives rise to the cellular condition. The mechanism of the cellularization has been investigated in the number of angiosperms, accounting the Newcomb and Quebec in 1973. Stellaria. So, the cellularization is brought to about the brought about by the fusion and the free growing wall. So here the cellularization in the iberis and facialis occur by the forming the mechanism, whereas it is the latter type of the paper somniferum and the allium crystallosa. The origin and the extension of the initially formed freely growing wall in the both type of the cellularization controversially both on the walls arise the projections from the embryo sac as the cell plates. So associated with the phragmoplast between the nuclei and the microtubule are associated with the growth walls. Okay. So here, cytology of the endosperm. The endosperm tissue is basically triploid in majority of the plants to the derivation of the form of the fission product of the three haploid nuclei. That is the one sperm nuclei of the male gamete and fused with the two polar nucleus of the female gametophyte. So that's why here triploid condition is present in the endosperm. The number of nuclei contributed by the male gametophyte in the formation of the endosperm is constant throughout the angiosperm. So the number of nuclei contributed to the female gametophyte alters with the type of the embryo sac. So it is one in Inothera, two in Polygonum, and Endemion, Adaxia, Drusa, and four in the Pinia plumbago, Fritillaria, and Plumbagella. Here 6 to 9 in Peperomia and 8 in Acalifa indica. So here 14 nuclei present in the faciolus and the polyoid endosperm also according to the post-fertilization tissue. Okay. So here the endosperm tissue also exhibits the high frequency of the polyploidization of its cells during the development. So here while the workload, the cytology of the species of what is that one? Here Nothoscordium and Ziphanthus. So here studied by the cytological flowering plant. So in Thesium alpinum, the polyploid of the endosperm hastorum is up to 384 and highest polyploid is represented by the area maculentum, whereas here 2000 here 24,576 nucleates are present. Okay, so in Dendrophthia, all of these cells are matured endosperm shows the remarkable stability in their chromosome number and rare diploid condition. Endomitosis and nuclear fusion are some of the methods of polyploidization of the endosperm. In increase in size of the endosperm nuclei can result from the polyploid or collagen in the pedicularis, males respectively. The common mitosis abnormalities in the intestinal tissues are the chromosome bridges, logging chromosomes, spontaneous breaks of the chromosome, and fragment. Size of the nucleus and the number of nuclei per nucleus also vary greatly. The endosperm is generally non-chlorophyllous. However, some chlorophyllous endosperms is also occurred in the some species, Raphanus, Athalio, and Crinum, Viscum. These are having the some chlorophyll are there. Okay. So next one is the Aliran endosperm, Aliran tissue. The sterile endosperm at the maturity comprise the two different tissues are there. The outer alveolar layer is consists of living usually one layer of thick triticum and zeamase here 2 to 4 cell layer thick in ordinium. The endosperm is maturity type. So in rice, the alveolar cells are two types are there. That is the first one is second one is the dense cytoplasm era at the end of the. The alluran cells have thick wall and non-vacuolated cytoplasm. There are the interconnected by the plasmadesmata. The most prominent organelles of the alluran cells are the alluron grains, followed by the pyrosome. The main components of the alluron grain are protein, chitin, and phosphates and other carbohydrates. So structurally, the allurar grains possess two kinds of the inclusions besides the ground of the substances. That is the globoid is present with the globoidal cavities. They contain the protein and lipids. Other inclusions which are the 1 to 1.5 diameter. So the ground sections also contain the high concentration of the protein, but less than the protein and carbohydrate bodies. Functions of the endosperm. So what are the main functions here? The endosperm to produce the nutrition to the developing embryo. So here some different functions are there. In endosperm seeds, the persistent endosperm is rich in reserved food material in the form of the carbohydrates, fats, and protein. During the seed germination, these substances are digested and utilized for the growth of the seedling until the latter develops a green pigment and is able to manufacture its own food. The second one is the more important contribution of the endosperm is the nutrition of the embryo during the early stage of the development from the zygote to the globular embryo. Okay. During the early stage of the endosperm as a nerve tissue, nerve tissues do not show the specificity for its own embryo. The liquid endosperm or endosperm extract from the immature fruit not only the support to grow own embryo, but can also nourish of the embryos of other of the other angiosperms. So, example, coconut milk, corn milk, liquid endosperm of the horse, chestnut, and the walnut. So here, what happened here? The young endosperm is rich in the various growth hormones such as the auxin, cytokinins, and gibberellins, whose concentration decrease after the certain ages of the tissues. So gelatin, very potent cytokinin, which is extracted from the young endosperm of the males, GMS. So this is the immature endosperm is also potent for the including the division is highly differentiated and mature cells, such as those of the secondary phloem in carrot. The endosperm also regulates the crisis mode of the embryo development, removed from the influence of the endosperm. Very young and embryos fail to the mature, even it is the richest artificial nutrition media. Okay. So here some embryogeny and shows the precious germination in the cultures. At the time of the fertilization, the embryo sac has very little nutritive material. After the endosperm, the advances in the development it stores the enough food substances for adequate supply for the development of the embryo. In the majority of the angiosperms, the zygote divides only after the endosperm has reached a reasonable stages of the development. Even in the role where the zygote is divided before and simultaneously with the endosperm, latter soon supposed to the embryo in the growth. Generally, the embryo grows only when the endosperm develops properly. If the endosperm aborts they happen in many incompatible across, the growth of the embryo is adversely affected. In the absence of the endosperm, like orchidaceae, Prostomiaceae, and Traphaceae, the special provisions exist to ensure the nutrition of the developing embryo. So during its growth, the embryo depletes the surrounding cells of the endosperm of their content. In many plants, such as the legumes and cucurbits, the embryo consumes the endosperm completely before the seed maturity. Okay, so what are here? What we discussed here. So finally, the endosperm is a unique feature of the angiosperm, and it take up the particular portion between the new and old sporophyte. What is the summary here? This is a unique feature in the angiosperm. So it is absent in some families of the Orchidaceae, Podostomiaceae, and Traphaceae members. So here, the endosperm is the reservoir of the food material where it is present in the mature seed and is divided into three types based on the mode of the development. So here, you cannot see in the nuclear division are followed by the cell plate formation and large number of nuclei are seen in the embryo sac in nuclear type. In other hand, that is the cellular type. Here, what happened here? All the nuclear division are followed by the cell wall. Immediately, cell wall formation in the cellular endosperm. Nuclear type, here there is no cell wall formation. And the helobial type of the endosperm is an the intermediate type where the first division is similar to the cellular type and the wall formation, but subsequently the divisions are similar to the nuclear type. So here are not followed by any cell wall. And all these types, the Hastoria are formed at the latter stage, either from the microphylla or chalazal region, either both, which are helps in the drawing the nourishment to the developing embryo. The development of the endosperm is coconut is somewhat peculiar as the embryo sac cavity is filled with the liquid syncytium or coconut milk. The endosperm of the different embryo sacs fuse together and composite endosperm is formed member of the Loranthesis. The feature with the unique to the if the control the endosperm is irregular due to the own activity or by the activity of the seed coat, such as endosperm set to the ruminate type. And we can see in the families of the Annulaceae, Aristolochiaceae. So sometimes lobing and folding of the cotyledons occur together, the rumination, and result in the formation of the labyrinth seed as a Convenalaceae and Anacardiaceae family. So here cellularization of the endosperm takes place in the own way in the different taxa. So the polyploid of the endosperm were considerable. The highest polyploid here 24,576 nucleids are in Aram. The endomycesis of the nuclear fusion are common causes for the polyploidization of the endosperm tissue. A layer of the living cells known as the alluran layer is seen in some cells, and these cells store the protein, phytin, carbohydrates, and phospholipids. The most important contribution of the endosperm is provide nutrition to the developing embryo. Okay, the endosperm perform with the formation is very well by sorting the various type of the food material and growth hormones in its cells. So here, besides, it is also regulate the mode of the embryo development.